thank you so much, Stephen. That was a really kind introduction. Stephen always commented on me when I gave introductions to speakers that came to our seminar series or visited us. And it's nice to see that he's now the one giving those introductions. So for those who don't know, Stephen was a grad student with me at Barrow and has since moved on and is now a postdoc at UCSD. So it's a real pleasure to be here, and really thanks for this kind introduction and the kind words. And yes, as Stephen indicated, we're really interested in this specific subtype of frontotemporal dementia and ALS that is caused by the mutation of C90rf72 that I'm going to explain in a little bit more detail throughout my talk. And what I would like to highlight today, so we have several projects around this mutation since it's been discovered, and we have a really fun and interesting project that was initiated by a former grad student who is now also a postdoc just up the street from you guys at Ionis Pharmaceuticals on the role of astrocytes and how astrocytes might contribute to the loss of synaptic function in degeneration, in particular in this disease. These are just some disclosures where I'm a member of scientific advisory boards. Unfortunately, none of these memberships support any of my research, so this is just stuff that I do for fun. So let's get started. So the question that we're all facing in the field of neurodegeneration, and I heard from Stephen that a lot of you, okay, let me do one thing because I'm gonna close out my Outlook so that this is not gonna interrupt us. Hold on one second. I'm gonna stop sharing here because this is gonna be very annoying, and I apologize, I should have done that right away. All right, we're back in business here. Apologies, there we go. So yeah, so what we're facing is this degeneration in the brain, and this is an MRI that was taken from an individual that passed away from Alzheimer's disease. And you can see to the right that we see this severe degeneration, shrinkage, and the changes in the brain structure and function due to this disease. And what we're really all interested in, what's causing this damage and how can we prevent this damage from happening? And Alzheimer's disease is one of neurodegenerative diseases where we have, a defect and a degeneration in specific areas of the brain, but there's a lot of other neurodegenerative diseases, and depending on what disease we're facing, different areas of the brain are going to be affected by this degeneration. And you can just name any of these diseases, ataxias, for example, that are heavily affected in the cerebellum and lead to degeneration in the cerebellum. And then we have frontotemporal dementia, which is really focused on the frontal temporal lobes. But then we have also motor neuron diseases and Parkinson's disease that affect the specific brain regions. And we all try to understand what's causing this. The one disease that we're studying in the lab is this ALS FTD disease spectrum. And so this is really an interesting disease spectrum, or people now call it a continuum, between a motor neuron disease and a dementia, and in particular the frontotemporal dementia. So this is really a disease that was recognized obviously individually when it was initially found, where we have a motor neuron disease that affects muscle weakness, muscle atrophy. It's a fatal neurodegenerative disease that we don't have any cures yet. We have some symptomatic treatments, but we still don't have any way to stop this disease from happening in the first place or to stop the progression once it starts. And then we have this frontotemporal dementia, which is a different form of dementia, different from Alzheimer's disease in regards to it starts at earlier age. It's one of the most common dementias that happens under the age of 65. And it comes in different flavors, so to say. Frontotemporal dementia can lead to a behavioral disorder, but it can also impact language and speech. And probably the one most recent celebrity that has been diagnosed, and you guys might have heard about this, is Bruce Willis, that does have this language and speech type of frontotemporal dementia, and he was officially diagnosed with this disease last year. So we have these two diseases on the spectrum here. On the left, the pure ALS forms, and on the right, the pure FTD forms. But then we have this motor and cognitive dysfunction that happens in the same patient, where we have indeed individuals that are affected both by this motor neuron disease, but then also developing cognitive impairment, and some of this cognitive impairment is really very specific to frontotemporal dementia. There's different variations on how much an FTD patient is affected with motor neuron disease and vice versa. And so in addition to this clinical overlap that we have between this disease spectrum, there's also a very strong genetic overlap. There are mutations, gene variants that lead to pure ALS, such as SOD1, which was the first genetic variant that was identified to be causative for ALS. And then on the other side of the spectrum, we have mutations in the MAPT gene that encodes the tau protein that leads to pure FTD. But then we have these genetic mutations that are found sort of in the middle here that can actually lead to both either ALS or FTD or a combination of these two diseases. So we have families where one family member has pure ALS, another family member has pure FTD, and then some family members are actually affected by both. We don't quite yet understand why that is, and this is one of the things that a lot of people in the community are obviously trying to understand and to better get a grip on how this happens and potentially how we can prevent this from happening. In addition to this overlap in regards to the genetics and the symptoms and the clinical symptoms, there's also a very strong pathological overlap between those two diseases. ALS is characterized almost uniquely, up to 97% of ALS patients, independent of etiology, whether it's a genetic version of ALS or sporadic version of ALS, has TDP-43 pathology. TDP-43, as most of you probably know, is an RNA-binding protein that is usually located in the nucleus of a cell where it manages RNA processing events such as splicing, and is mislocalized in disease to the cytoplasm where it can aggregate into toxic aggregations. And both the loss of function and the toxic aggregation can actually contribute to the degeneration of the cells that are affected by this. We have TDP-43 also in about half of FTD patients, where we see the same pathology. TDP-43 is lost in the nucleus, aggregates in the cytoplasm. But the other sort of 45% of FTD patients have a pathology in the tau protein. And then we have a small percentage that actually has another pathology in another RNA-binding protein called FUS. So as highlighted by my title and also by the introduction that Stephen gave you, we're really interested in this one particular genetic mutation in the gene called C90rf72. The reason why we're really interested in this mutation is because this mutation is at this point still the most prevalent genetic variant in ALS and FTD. It was discovered by two independent labs in 2011, where they found this large repeat expansion of a G4C2 repeat in this gene, and they found it both in ALS patients and in FTD patients. And since then, because of the high prevalence, the community has been extremely interested in trying to understand what leads to the degeneration caused by this genetic variant, including our lab. Just to give you a little bit of a background, although I'm not going to talk a lot about these specific mechanisms, since its discovery, we've identified four major pathways and mechanisms that contribute to what happens in an individual that has the C9 mutation. Again, this is a hexanucleotide repeat expansion, a G4C2 repeat that's found in the intron region between exon 1A and exon 1B. And there are different variants of this C9 gene for now, and I think we might be at 3, but some people had speculated earlier whether there might actually be additional variants of C9. But let's say there are 3 variants that can lead to both a long protein as well as a short protein. And the repeat expansion is only found in 2 of those variants, in variant 1 and in variant 3, within this intronic region. What happens as a consequence of this repeat expansion are 3 major sort of cellular events. Firstly, we have what we call a haploinsufficiency. Because of the inclusion of this repeat expansion, we're actually not in the allele that carries the expansion. We cannot translate into a full protein, and we do see about 50% of C9 protein translated due to this mutation. So there could be a loss of function of the C9 protein that contributes to the degeneration in the cells affected. Another pathway that we have known about through other repeat expansion disorders, such as myotonic dystrophy and spinocerebellar ataxias, what happens when you have repeat expansions? These repeat expansions, the RNA often forms and clusters together in so-called RNA foci. And as a consequence of these foci, you actually get sequestration of other RNA-binding proteins that are in the nucleus of the cells. And as a consequence of that sequestration, you get a loss of function of those RNA-binding proteins. And that is what we call an RNA-mediated toxicity. Unfortunately, we have not yet identified a specific RNA-binding protein that might be contributing significantly to the degeneration. This happened in myotonic dystrophy, for example. There's a muscle-blind protein that we know is sequestered to foci of the repeat expansion in myotonic dystrophy. And the loss of function of muscle-blind is one of the major drivers of degeneration in myotonic dystrophy. Unfortunately, we have not found the muscle-blind-like protein in the case of C90rf72. And then the third major mechanism that happens due to this repeat expansion is this formation of so-called dipeptide repeat proteins or products through a very interesting translational event called RAIN translation, or repeat associated non-AUG translation. So what happens, you have these really long repeat expansions. I forgot to say, these repeats can go up into the thousands. So they're really, really long. And instead of having a typical canonical translation that happens on the ATG start site, this repeat can actually be translated throughout the repeat without this AUG start site. And as a consequence, we form so-called dipeptide products. And there's 5 of them. There's actually 6, but 2 are being translated both from the sense and the antisense strand. And depending on the composition of those DPRs, they can be more or less toxic to the cells that they are generated in. And this is yet one of the other mechanisms that we think contributes to the toxicity in this repeat expansion. And then last but not least, and I already mentioned this, patients that have the C9rf72 mutation also have TDP-43 pathology. So again, you have the translocation and the loss of a nuclear protein that is highly important in RNA processing events that now translocates into the cytoplasm. And so we have a loss of nuclear TDP-43 and the gain of cytoplasmic toxic accumulations through these aggregates that are formed in the cytoplasm. So with all this knowledge, we've done a lot of, by we, really scientific community, has identified many, many mechanisms that we think are attributing to the degeneration that we see in this disease. And you can pick your favorite pathway and your favorite flavor that you're studying: DNA damage, autophagy, RNA processing and splicing. All of these events have been associated and have been found to happen in C9 ALS FTD. But what we still don't understand quite is what actually initiates this neurodegeneration, right? So all of these, I shouldn't say all, but many of the processes that have been identified are probably downstream processes, right? This is what happens once the mutation does X, that then leads to these dysfunctions that we see in the cells. But what is truly X? Like, what really initiates this? And that is obviously the million-dollar question, because we would all like to go upstream in the pathway activation, because if we fix something further upstream, we can prevent all of these downstream events from happening. And so I always like to get the lab to sort of take a step back sometimes and look at the big picture, right? So what really happens in the brain and how does a brain function? I call it brain function 101. So what happens for a brain to function is that the cells have to communicate with each other, right? I don't have to tell you this. This is really mostly, I use this slide often when I speak to patients and patient families. But we really know that synaptic function is critically important for the brain to function. And just to sort of keep in mind, and I think we often forget about this, so we have about 100 billion neurons in the brain. And with those 100 billion neurons, and depending on the number, I actually Googled it yesterday to see what the current number is of how many synapses the brain actually has. Let's just say it's over 100 trillion synapses that we have in the brain. So these synapses are really, really critically important. And no surprise that we've noticed, not just in ALS and FTD, but really also in other neurodegenerative diseases, such as Alzheimer's disease, that there is an incredible significant loss of dendrites and synapses early on in the disease, really still in the preclinical phase, while there is a start of inflammation and protein aggregation. But this event happens really early, and we believe that this is truly a critical event that happens before we see any of the actual sort of downstream effects of the degeneration of a neuron, right? First, the neurons don't connect to each other anymore. And then the neurons might decide, well, if there's no connection there, why am I still here? And then there's other things that happen, obviously, that could then lead to the more severe degeneration and the ultimate loss of the cells in the brain. So let's go back and look at this synapse a little bit more closely, just to remind ourselves, and especially for those of you that are not thinking synapse at all times, which is what I do and my lab does. But let's just really look at this. So we have these connections in the brain, and the connection occurs at these synapses and often at these dendritic spines. And we have the presynaptic terminal and the postsynaptic neuron, right? So the information comes presynaptically, gets received by the postsynaptic neuron, and is then transmitted further down. And a lot of cellular events obviously happen when this connection occurs. So what we also know now is that this doesn't happen in a vacuum, and we really need the participation of glial cells in order to make this synaptic transmission very efficient, because we need efficient transmission for this to happen quickly. For me to be able to do what I do right now, to speak, hopefully faster because I'm running out of time already, it has to happen really quickly. And it's really the glial cells that are surrounding the synapse that provide this efficient and fast synaptic transmission. And that is often really overlooked. And so because of that, there's a number of people in my lab that are working on this aspect of glial contribution to synaptic function, and in particular, obviously, to the synaptic dysfunction that we think occurs in FTD, ALS, but other degenerative diseases as well, and we think occurs early on. And the project that I'm going to talk about is really a project that was spearheaded by Lynette when she was a grad student in my lab, and is now continued by a new postdoc in the lab, Robert Colbrick, who's going to follow up with the studies that Lynette initiated. But I cannot talk about glial cells in ALS or FTD without mentioning work that has been done by a quite prominent investigator at UCSD, Don Cleveland, who really all in the early 2000s really sort of put this seed in our brains to say that in case of ALS, you know, the motor neurons are dying. But he immediately said that, well, the motor neurons are not dying just by themselves. There is really an important contribution of other cell types that contribute to this neurodegeneration, such as the microglia and the astrocytes in one shape or another. And he went further on to show this in some really elegant studies. There was no CRISPR back then, so this was hard work, you know, introducing cell type specific gene mutations in the different cell types of, at that point, a very common mouse model of ALS, the mutant SOD1 mouse, which was really the first model that we had for ALS, where he showed that depending on where he introduced the mutant form of SOD1, there were different outcomes in the disease. And we don't even have to go through the details of this. It's just to highlight that Don really, you know, even back then in the early 2000s already told us that the glial cells are actually really important and they contribute to the neurodegeneration. Yet most of us sort of, look at these diseases very neurocentric. And so here, I want to tell you one of the stories that we did to look at the role of astrocytes in particular. And you can see here that astrocytes play a lot of important physiological functions. They provide trophic support. They take up neurotransmitters, in particular glutamate. They're really important to maintain the glutamate balance in the synaptic cleft with activity of glutamate transporters, and so on. The most important part that we're interested in, because we're very focused at the synapse, is its role in synaptic formation, maturation, and function. And there certainly has been indication that there is, potentially a contribution of astrocytes to this decreased synapse formation that I showed you earlier, and thereby an altered neuronal activity. So a lot of this has been done in spinal cord astrocytes. ALS is a motor neuron disease, and a lot of people focused on the fact that, just like I showed in the papers that came from Don Cleveland's lab, that motor neurons are dying, but they're dying with the contribution of spinal cord astrocytes. Well, astrocytes in the spinal cord are actually very different from astrocytes in the frontal cortex. And so what Lynette wanted to uncover, and what she wanted to look into, is, well, what happens with the cortical astrocytes? Because we have a disease spectrum that also has frontotemporal dementia, which affects the cortical aspects and the frontal and temporal lobes of the brain. So what happens to those cortical astrocytes, and how might these cortical astrocytes contribute to the cortical degeneration that we think contributes to this cognitive impairment in this disease spectrum? Lynette took a very sort of multi-omics approach by doing this, and she used iPSC differentiated cortical astrocytes that came from C90rf72 ALS FTD patients. And all of this was done with a really close collaboration of Dr. Van Kuren Jensen and Dr. P30. Both of them at that time were working at the Transgenomic Institute, or TGen, in Phoenix, right around the corner from where we are, where they helped us to perform a very thorough transcriptomic and proteomic analyses of these iPSC cortical astrocytes in order for us to start understanding whether there is some cell autonomous dysfunction in these astrocytes caused by the C9 mutation and the G4C2 repeat expansion. So this is just a flowchart on how Lynette generates these iPSC cortical astrocytes. We go through the generation of neural progenitor cells. That is important because what we're doing right now is we're also using these NPCs to make cortical neurons and to do co-cultures of these neurons. And I unfortunately don't have data on this yet, but this is what we're currently investigating now that we understood what happens sort of in the monoculture of these astrocytes. Lynette took beautiful images of these neural precursor cells or progenitor cells. They have all of the markers that NPCs should have. Again, these are NPCs that can become both astrocytes and neurons. Here's some staining for nestin, for PAC6. She really confirmed that these NPC markers are all present and that these cells form the very typical rosette morphology before she then differentiated these cells into cortical astrocytes. When she did this, she wanted to make sure, did I really make cortical astrocytes? Because I told you before, a lot of work was done with spinal cord astrocytes. So she looked for regional markers that are expressed specifically in cortical astrocytes. And one of these markers is F2C, which is, as you see here, highly expressed in your immunocytochemistry that you did. So you can see here the cortical astrocytes to the right, and here she has some spinal cord astrocytes to the left, where there is no MEF2C expression, yet there is strong expression in the cortical astrocytes. And in converse, she looked at a spinal cord marker for spinal astrocytes, which is HOXB4. And you see that very strong expression in the spinal cord astrocytes, but you don't see that expression in the cortical astrocytes. She confirmed this then once she had her transcriptomic data and her proteomic data. She confirmed again the expression of MEF2C on the mRNA level as well as the protein level, using her omics data to support her immunohistochemistry that she did. Do these cells make typical astrocytic markers? She looked for many of them. Here's an example of aquaporin-4, S100 beta, and ALDH1L1. Again, she started with immunocytochemistry, and she looked for the expression of these markers. There was no difference between control and disease cortical astrocytes in regards to the expression of these markers. So both healthy control and diseased cell lines actually generate very typical brain astrocytes. And again, she confirmed this data with her proteomics and her transcriptomics data that she did later on, confirming that there is no significant difference of these marker proteins and genes between ALS or ALS FTD and controls, and all of them are really present. So we were dealing with, astrocytes that are of cortical identity, and they express the typical markers. And then she did a full omics analysis. And due to the time restriction, I'm going to put what probably is work of almost a year in one slide. And she's probably going to kill me if she sees this, because a lot of work went into this. But really, the bottom line is that she got, a significant presence of genes and proteins. She saw changes, differentially expressed genes and differentially expressed proteins, with about 44 of the genes that were dysregulated either down or up. Affected pathways were mostly cell adhesion and ion transmembrane transport, which was really interesting for us. She also found significant numbers of differentially expressed proteins out of the 9,000 proteins that we were able to identify using mass spec analyses. And in addition, we also looked at phosphorylation, and we looked at phosphoproteins, and she found out of 8,000 unique phosphorylation sites that we identified in the data set, that 69 of those were abundantly phosphorylated. So there were changes in the phosphorylation of 69 sites. And then with that information, we were able to sort of indicate what kind of kinases might be differentially active based on the phosphorylation sites that we found. And about 24 of those kinases were differentially active, and 9 of them overlapped actually between the proteome and the phosphoproteome, which was really interesting. And one of the major pathways that came up when we did the pathway analysis of all of the omics was synaptogenesis, which for us was a really, sort of a big smile on our face, because that's what we expected to happen. And we were really excited to see this. And so when you do these multi-omics data, I'm always worried, because you have these big data and you're like, oh my God, what am I going to get out of this? It's always nice to confirm something that has already been shown. And in our case, one of the nice confirmations that we found was a change in the connexin 43 protein that has been associated with ALS for a long time. And we can see nicely that we do see an increase in connexin 43, which is truly what other people have shown in other forms of familial ALS. And she confirmed that both on her proteomics data. And while it didn't reach significance, but you can see that there is a trend to also see increased gene levels of connexin 43. And she confirmed this further when she ran a Western blot analysis on her iPSC cortical astrocytes. So that was a nice confirmation on that the data that we have can actually confirm something that has already been described. So now, how does this repeat expansion now affect neuronal synapses? I've told you a lot about these synapses and that we're really interested in them. So let's look at this in more detail. So there are different ways that astrocytes can support a neuronal synapse. On the left, we're talking about an astrocyte synapse direct contact that can regulate the structure and the function of the synapse. And these include proteins like ephrins and ephrin receptors, astrocytic neuroligands and norexins, as well as there's different flavors of the ephrins that are listed here on the left. And then on the right side, we highlighted a few of the secreted proteins that are known to contribute to synaptic strength just by being released and then by binding and stabilizing what we call this transsynaptic bridge that is built between neuronal norexins and neuroligands, and that really keeps the synapse together. And these 3 proteins that we looked at in particular were a protein called SPARC, one that called SPARCL1, and thrombospondin. So let's go through this one at a time. So neurexins and neuroligands, as I said, are not only found on neurons, but they are astrocytic neurexins and neuroligands that really sort of help keeping the astrocytes close to the neurons. And astrocytes highly express certain subtypes of these, neurexin 1 and 2 and neuroligin 1, 2, 3. And they're really, as I just said, sort of contributing to this astrocyte neuron communication. The other flavor of proteins and ligands that are known to stabilize the synapse by direct contact are ephrins and ephrin receptors. Again, there are different classes and categories, and these receptors and ligands have been associated with ALS before, mostly in the context of SOD1, but not yet for C9. So when Lynette focused on the neurexins and the neuroligands, this is what she found. You can see here on the top is the proteomics data, and on the bottom is the transcriptomic data that she got. And here are the different neurexins. So this is neurexin 1 and neurexin 2, and you can see again the proteome and the transcriptome on the bottom. And here we have neuroligin 1 and neuroligin 3. And really only neurexin 2 and neuroligin 3 showed a significant difference. And most of these differences were really found for our data set on the protein level. The transcriptomic, as you can see here, always went in the right direction, but for some reason did not reach significance. But the protein data were actually really robust and showed us a significant difference between the controls and C9 with a downregulation of both neurexins and neuroligin 3, suggesting that with a reduction of these proteins and anchors, that this might indeed contribute to loosening up the synapse and reducing the synaptic strengths. Let's look at the ephrins. Again, we have the protein on the top and the transcriptomic on the bottom. We have ephrin B1 and ephrin B2 and B3, and we were only able to detect one at the ephrin receptors on the protein level, which is ephrin A2. And what Lynette showed is that, again, there is a significant downregulation of ephrin B3, which is coming from the astrocytes. Interestingly, there was an increase in the levels of the receptors. One might speculate, and we have not followed up with these hits yet to really see what the cause and what the reason for that is, but one might speculate that there could be overcompensation. If the ligand is missing, that the receptor might actually try to overcompensate to look for its partner and just generate more. But this is just speculation, pure speculation, and we really don't know yet what's happening. So just to give you a summary on what happens in regards to astrocyte synapse contacts, we do see decreased levels of neurexins and neuroligands, in addition to decreased levels of ephrin B3, and we do see an increased level for ephrin A2, which is the receptor for the ephrin ligands. So now let's look at these secreted proteins and the secretome of astrocytes and of proteins that have been known to be involved in synaptic strength. They are the thrombospondins. Again, this is a glycoprotein that's supposed to facilitate cell-to-cell and cell-to-cell matrix interactions. There is SPARC, which is also a glycoprotein that is supposed to be shown involved in synapse elimination. And so SPARC is really an antagonist of SPARCL1, which is yet another glycoprotein. So what happens is that SPARCL1 and SPARC can actually bind to each other. And when SPARC binds to SPARCL1, SPARCL1 can no longer support the neurexin neuroligand transsynaptic bridge here, and thereby destabilizing the synapse. So these usually go in the counter direction. Often when SPARC goes up, that means SPARCL1 can no longer be active. We didn't find any changes in thrombospondins and SPARC, neither on the RNA level nor on the protein level. But we were really excited to see that there was a significant difference in this astrocytic SPARCL1, which is also known as Heben, which is decreased in the proteomics data. Again, there's a hint at a significance in the transcriptomic data. It did not reach significance. Lynette very quickly validated this by looking at Western blots of the cell lysates. So she, in addition to the omics, proteomics analysis, she ran Western blots and she confirmed that there's a decrease in SPARKLE1. And most interestingly, she also looked at astrocyte conditioned media, which is where we should find SPARKLE1, being it a secreted protein. And when we did Western blot analysis, again, she saw that there is a decrease in the amount of SPARKLE1 that's being secreted in the supernatant. So we were really excited about this because Heben, or SPARKLE1, and SPARK have definitely been proteins that have been associated in neurodevelopment to be critically important with synaptogenesis in the CNS. All of these studies have been done in mouse. Very little information has been gathered in human, and very little to no information has been gathered whether any of this is actually dysregulated and dysfunctional in disease and could contribute to these synapse losses. But a lot have been done, especially with Ben Barris's group and Kala and Nicola Allen. Those are the three major players that really pushed the field forward in regards to the role of Heben or SPARK or SPARKLE1 and SPARK. And in regards to if there's lots of Heben and little SPARK or lots of SPARKLE1, then you have lots of connections of astrocytes and lots of synapse connections. But if you have more SPARK than Heben, then this is inhibiting and you see a destabilization of the synapse. So we looked around and to see whether anybody else had looked at SPARKLE1 in regards to ALS and FTD. And there was indeed a paper that was published to look at the proteome actually across the ALS FTD spectrum. And again, while it wasn't significant, but they did show when we looked at the data set and Lynette analyzed their data set that there is a trend to a decreased level of SPARKLE1 in this human brain tissue analysis that we got from this study that was published before. And again, this is they actually distinguished between ALS groups that were negative for C9, so non-C9 mutants, and C9 mutants. And in both of them, you actually see that it looks like there's, although not significant, but it looks like there is a trend to a loss of SPARKLE1. Interestingly, also, again, Ben Barris's group looked at reactive astrocytes. So they defined reactive astrocytes way back, and they actually looked at reactive astrocytes, or they created reactive astrocytes in an SOD1 mouse model. And when they did this, they showed that SPARKLE1 mRNA expression is significantly decreased in those, what they call reactive astrocytes, and they call them A1 astrocytes. We don't really use that nomenclature anymore, but reactive astrocytes is still something that we say, or more of a reactive state that the astrocyte goes through. And I'll come back to this later with some of our own data. There was also data on CSF proteomics, and this was actually done by my colleague, Dr. Bowser, in 2015, where they looked at the proteins that were found in CSF of ALS patients. And you can see that SPARKLE1, interestingly, and this is sporadic ALS, again, so this might not be specific for C9, there is a decrease of SPARKLE1 when compared to healthy controls or other neurodegenerative diseases. So that was really sort of interesting for us and confirming that SPARKLE1 might indeed play a significant role in this disease. And more interestingly, that SPARKLE1 could potentially actually be an early biomarker of disease if we can detect it in the cerebrospinal fluid. And then last but not least, the other supporting data that we found was there are SNPs in SPARKLE1 that were identified. And supposedly, those SNPs in SPARKLE1 accelerate actually symptom onset for Alzheimer's disease. So you can see here for the individuals that have the SNPs, there is a very strong correlation with memory loss when they did behavioral analyses of these patient populations. So that was sort of really interesting as well, that there could actually be genetic variants of SPARKLE1 that again indicate that with the loss of function of SPARKLE1, there is a contribution to cognitive impairment. So luckily, we had a single nuclear RNA-seq data set that we recently generated that was done by another member of the lab, Lauren Giddings. again in collaboration with Dr. Van Kuren Jensen, where we looked at single nuclei RNA-seq analyses of C9 patient brains. And we differentiated between C9 ALS, ALS FTD, and FTD in comparison to controls. And we looked at the frontal cortex as well as the occipital cortex and did a very sort of old-fashioned at this point single nuclei RNA-seq analysis. And while we were focusing on something different with this analysis, we obviously get our cell clusters. And you can see here that we clustered in, I don't know how many, 11 or 16 different major clusters that were identified, and they were associated with cell type specific markers, including the astrocytes down here. And you can see them right here. I always think this looks like a dog head, so I'm going to go with it. Could look like astro. So what we did is we pulled out the astrocytes because that's what we're interested in. And what we did is we pulled all of the astrocytes from all of 3 C9 patient subgroups. And you can see that they mix very nicely. So there's not really a disease-specific subgroup that represents one cluster more than another. And when we further now dove into this and we decided to make subclusters of these astrocytes, we sort of came up with 4 major clusters. And interestingly, so we pulled out some of our major players that we're really interested in. And what struck out here is that we have these 2 clusters, cluster 0 and cluster 1. And cluster 0 and cluster 1 seems to be almost at all times opposing in the level of gene expression of major players. So here's Sparkle 1, our favorite, which is significantly down in cluster 1, yet significantly up in cluster 0. That goes along with a loss of norexin. You can even see neuroligin 3 is also down. It's blue versus green. It's not as highly expressed, but it's still, significantly changed. This is EAT2. EAT2 is the glutamate transporter that is important to remove excess glutamate from the synaptic cleft. Interestingly, again, we have a loss of EAT2 in those, what I now call reactive astrocyte cluster 1. And we have an increase in GFAP, which is, again, another indicator for activity and reactivity of astrocytes. So we thought that we really found actually 2 astrocyte clusters that are sort of opposing roles that we now call homeostatic and reactive. And this is just sort of a unique gene collection. Again, this is a lot of data. I just wanted to highlight, again, the ones that I already highlighted. EAT2, and norexin 1, which is down in cluster 0, but it's up in cluster 0, but down in cluster 1. Unfortunately, they changed this up on me. This is what the bioinformaticians do, just to keep me on my feet. And again, down here, we have now cluster 1, which is the reactive cluster, where we have an increased level of GFAP, but we have lower levels of the other genes that I just highlighted. So that was super interesting and sort of really got us to think about looking more into those reactive astrocyte clusters and to try and understand whether we can figure out overall, what potentially triggers this reactivity in these astrocytes that we sort of, identified through the omics data from our iPSCs and then confirmed in the brain tissue. Again, these are volcano plots. You pick your favorite, and these data are freely available online. So these are all of the data from the single nuclear RNA-seq that are available online through our publication. And we divide it now up between ALS, ALS FTD, and FTD because we're really curious also whether there's any disease specificity among these clusters. And we did this similarly for the homeostatic astrocytes, which had a ton more changes in the differentially genes. I have no idea why. Sort of interesting again, we're just in the beginning of understanding this. All of this, has really not yet been validated, and a lot of bioinformatics work now needs to go into this for us to thoroughly understand what's really happening with these dysregulations now that we identified what we think are relevant astrocytic subclusters that could lead to this synaptic dysfunction. So, but how is this really happening? And so Robert now is tasked to figuring this out. And so he's really trying to figure out what triggers the change from a homeostatic astrocyte that has lots of SPARKLE1, lots of E2, neurexins, very little GFAP, that then turns into this reactive astrocyte where we just have the opposite happening. And how, obviously, the most important question, how does this contribute to neuronal function? And so the way we're starting with this, and this is really hot off the plate, so I just wanted to throw it in to also recognize Robert's work on this. So we're knocking down SPARKLE1 in iPSC astrocytes, and we already have this ongoing, but I don't have any data yet. And we're co-culturing these knockdown SPARKLE1 astrocytes with neurons, and this is all just in controls, to see whether indeed the loss of SPARKLE1, and we also, when we knock down SPARKLE1, we do see less of SPARKLE1 in the supernatants, which is really important because it's a secreted protein. So these are ELISA assays that we ran. We now switched over to ELISAs. They are much more sensitive and give us a much better quantitative readout. And so what's cooking right now, and I, again, don't have the data yet, is the co-cultures between the SPARKLE1 knockout cortical astrocytes with the neurons to see whether if we indeed knock out SPARKLE1, is that sufficient to lead to a dysfunction of neurons in regards to their synaptic function, their excitability, and their number of synapses that they form in these in vitro structures. So this is where we're at right now. Again, this is all unpublished except for the single nuclear RNA-seq data. We're super excited about this. Just to sort of conclude, we were able to make cortical astrocytes. They look like cortical astrocytes. I didn't have time to go into this, but they do have C9 phenotypes. They have foci. We have a lowering in the C9 protein. These data, the C9 phenotypes, are very difficult somehow to detect in iPSC cultures, and this is happening across the field. Here's just an example that our data looks very much like a data in spinal cord astrocytes that were done from C9. We do see differential gene and protein expression in these monocultures. We're going to repeat some of this in co-cultures to see whether we actually get an exacerbated change of the differential expression of genes and proteins. And we're also looking, and that's a project that another grad student is working on, how microglia activation actually changes astrocytic reactivity, because we think that there's a very important crosstalk between microglia and astrocytes that could also contribute to the change of a homeostatic astrocyte into a reactive astrocyte. We confirmed some of the known players, which was always comforting to see. And we think that SPARKLE1 is one of the critical players that significantly contributes to this synaptic dysfunction that occurs early in disease. And we're hoping that maybe SPARKLE1 could be an additional biomarker that we could utilize because it is detectable in CSF. And now what we're trying to do is look for existing CSF samples. A lot of people are now doing longitudinal studies, collecting CSF from ALS FTD patients. Can we measure SPARKLE1 in those longitudinal samplings that we have and see how early does it actually come up? And is it a true early indicator of degeneration in this patient population? I want to thank my lab. We had our first lab retreat this year, and we went up to Flagstaff, which was wonderful. They convinced me that I had to go on an obstacle course hanging, by ropes for my life, not falling off. But we had a lot of fun. And thanks to Lynette, who really did an incredible job in putting the story together and for providing us now with a really cool follow-up study to look at how these astrocytes really affect synaptic dysfunction and how we can utilize this potentially for biomarkers and or even therapeutic treatments. Thanks to all of the collaborators. I mentioned the collaborators that mostly helped with the project that I presented today from TGen. Mostly in this case, but we have collaborators and actually the Mayo Clinic in Florida helped us with the DPR measurements. Tanya Gendron was the investigator that helped with her ELISA for DPRs. None of this would happen without funding. We were very fortunate. Lynette actually received a supplement award when she was a grad student to support her studies on this project. So we're super excited about this. And with that, I'm happy to take questions and can't wait to hear whether you guys have any good suggestions on where we should go with this. Thank you.